Okay, hi class. Glad you're here. I think it's time to start. Before we get into today's class stuff, can somebody remind me what's happening for lab this week? Lab this week. Anybody know? Yeah. Easton, tell me really loud. Yeah. Do you have to be in lab to do this? Yeah, you're right. Yes, fair. If you need to be in lab, great. If you don't need to be in lab, also great. If you need stuff for this, you should you need to tell your lab instructor person if you need stuff that comes from lab. Like if you need 2 carrots and an egg to do your lab, that's on you. If you're like, I really need the enzyme amylase and I'd like to use some Benedict's reagent, ask your person, okay? Everyone's cool with this? Whether or not you're in lab or not in lab, you need to do your lab. It is your self-design experiment. Questions about this? Comments? You should, I believe you got, I don't always get all the emails you get. I think you got a long-winded email about this from Robin. Yeah, being like, do the things. Questions about this? Sweet. So that's this week for lab. It's very exciting. Okay. Anybody remember what's happening on Friday? This Friday in class? You so know. I think. Nobody? Yeah, Raven. Yeah, we get an exam. Yay, right? Yeah, so I'm guessing you just got done taking a whole bunch of midterm exams? No? They're all this week. Okay, well, see, if you, we go through it and we're like, okay, a third. And this is where it lands. If people only do like one massive midterm, it lands like a week and a half ago. But if people split it up into exam 1, exam 2, final, which is what a lot of people like to do because otherwise it feels like one test rules them all. If you have one bad day, you're toast. Anyways, all this to say, it's this Friday. It's gonna be online again, like the other one. I was asking the 9 o'clock class how the format was working for you guys. How is the format working for you guys? It's okay? I realize it's kind of tight on time, and I feel like I have to have it be tight on time. So people can't just look up every single answer. Is there any interest in having the multiple choice thing still be super tight on time, but then you open up an entirely different exam tab? I mean, it's still all the same exam thing to do. That's then the short answer ones that you have longer to do. Interest in that, or is that just one more thing and it adds more time? Anybody for keeping it exactly the same way? Anybody for having one more thing to push on the buttons, but slightly more time on the short answer part? I still have to keep it within the hour. So do people see the difference there with what's going on? So version A is presumably, version A would be something like, okay, you have 60 questions, 10 of them are short answer, but the first 50 you have 40 minutes to answer those, but you've got 20 minutes for the short answer ones, right? Or it's just all in there in one big blob. No, well, I'll make it and then see how it's going, and yeah, I don't know. I like grading your stuff online. I will not miss doing online exams once we're back to sort of normal, because it just, I feel like I have to be sneakier with them. Okay, so today's class, we're talking about the cell cycle. And this is kind of a different feel of a topic than the last couple. So the last couple, things like metabolism and cellular respiration and photosynthesis, a lot of people knew some about it. Most people hadn't actually gone through like glycolysis in depth in high school, right? The cell cycle is a little bit tricky because either people have gone through it in nauseating detail in high school and like had to act it out and had to be the metaphase plate or whatever, or they've never heard about it before and they're like, it's one more thing I've never heard about. Okay? So if you're somebody who's heard this so many times, a few deep breaths, let it wash over you, right, as you learn stuff. If it's completely new, also cool. But here's where we're at. Okay? You might have noticed, I don't know how dutifully people are reading the books, you might have noticed we've skipped a chapter in the book. So, up until now we've been reading every single chapter. This past, so going from photosynthesis to here, the cell cycle, we skipped cellular communication. The brief take-home message from that chapter was, hey, guess what? Cells exist. Cells listen, not like with physical ears, but they listen to the environment. So they respond to outside signals. They can send signals to other cells. Cells can talk amongst themselves and respond to the outside environment. I mean, there's more in the book, but that's the gist. Is everyone okay with this? Okay, sweet. On to the cell cycle. So, things that are alive need to make more of themselves. And this ability to make more of themselves is basically the definition of what it means to be alive. You can make an argument for like, is fire alive then? Because it can spread, but no, it's not alive. But like, if you have an octopus, your octopus needs to be able to make another octopus, right? There can be multiple octopuses involved, right? A tree needs to be able to reproduce to make another tree. Moss needs to be able to make more moss, right? And it's not just like sexual reproduction, right? The individual organism also needs to be able to make more of itself, right? Or typically does. When you started out as a little zygote, one cell, something or some things has happened between one cell is going to be you and you right now, right? Like something went down in the middle there. Is everyone okay with this? Yeah. So cell division is going to set up so much. And cell division, in my personal opinion, is the most boring topic we talk about because like, yep, you need to double stuff and bloop, right? It's important, it is kind of boring, it's good for you though. But this sets up some of the really interesting stuff, the how come genetics works the way it does, and how come you don't spontaneously grow extra arms, and how come cancer is a problem, how to stop it, that kind of thing. It sets up a lot of the foundational stuff. Sorry, this is foundational, it sets up a lot of the more interesting stuff. Okay? So, here we are. It is kind of pretty. So, like, don't memorize anything here, it's pretty. Another one, we'll talk about what's going on here. Ah, it's not changing. Thank you, Fi. Tell me, somebody. I've been pointing to slides in my, oh my goodness. Sorry, people, you're right here. I didn't realize it was frozen. I was checking my email. Da-da-da, 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 pretty. Everyone's here? Tell me if this ever happens. I'm like, I have no idea what you're talking about, woman. Okay? We're good? It's pretty right here. Great. Thanks. You are the most chill class I've ever had. Which, I'm not sure if that's a good thing or a bad thing, but it's kind of nice, but it's also kind of, anyway. You're also a really polite class. Which is kind of nice, too. Also, while I'm saying nice things about you, and it's true, you know that for many people, today is a holiday? Props to, like, still being here and trucking along. Yeah, anyways. I enjoyed being undergrad. I don't envy any undergrads this particular year. I'm glad you're, like, hanging in there. And I realize in your calculus of what makes sense for you, it probably makes more sense to be here and be moving forward with your life plans as opposed to like just be spinning your wheels. But it's got to be a little bit rough. Anyways, yay for you all. The pictures are pretty and yeah, sheesh. Okay. While we're at okay. If you have a unicellular organism, how many okay, dumb question. Unicellular organism, how many cells are in this organism? One, right? Unicellular organism. Boom. Its one cell is its whole it. Right? Okay. If this unicellular organism splits and the cell divides, now there's two cells, how many unicellular organisms are there? Two, right? You're like, yay, boom. Okay. Cool. So if you've got an organism that's one cell, cellular division is reproduction, right? Now there's two of them. If you've got a multicellular organism, something like you, or a pine tree, or a sea cucumber, or whatever, right? There are other things going on for cell division. Why do you need cell division? Yeah. So sometimes cells die, right? Um, gross facts, not gross facts. You know, like when you dust in a house, most of that dust. So some of it is dirt from outside, but typically most of it is chunks of the people who live there. And most of it's like old skin cells. It's kind of gross. So yeah, there's that. So lots of news coming up, right? Or um, things die. They get old, they get worn out, cells need to be replaced. So there's that. Also, you're gonna need, so if you start out single cellular as a zygote, a series of things need to happen until you're now with your however many billion cells you have. So you need to develop from a fertilized cell. And also if you actually physically grow. So as you're developing, during development this happens, during growth this happens, during repair this happens. So cell division needs to keep happening. The cell cycle, you might hear this sometimes, the cell cycle is the whole like series of events that the cell does. And cell division is a very small part of that whole thing a cell does. But whether in reproduction or for doing tissues or for growing and developing, you're expecting cell division to go down. Okay. We're going to go over two concepts today. I've called them thing one and thing two. Thing one is most cell division results in genetically identical daughter cells. People talk about what's called meiosis, and they talk about mitosis. And I think people often think that they're like both equally common. They are so not equally common. Most of the time when cells divide inside of you, you're not actively reproducing. Is that fair? Great. So most cell division is going to result in genetically identical daughter cells. It's called mitosis. It's going to have genetic information that's the same. So let's say a specific portion of your liver gets damaged. Some cells in your liver are going to grow and divide, and now you have cells back in your liver, they're doing just great. Okay? Sneaky question. Are the cells in your right earlobe, do they have the same genetic information that the cells in your left big toe have? They have the same genetic information inside that cell. Yeah, they do. I thought I was being sneaky. I wasn't being sneaky. Okay. They have the same genetic information. The information for how to be you, right? Are people okay with this? They do different things, right? Like, or the cells of your eyeball, they're doing different things than the cells of your bones, but they have the same information there. Is everyone with me on that? We're going to go for a half-baked analogy here. Have you ever, like, you have a bunch of photos on your phone, and then you're trying to back them up to, say, Google Photos or something, right? You copy all those photos and you put them someplace else. Yes? Is everyone cool with this? You've done this before? Okay. As you do that, you should now have a copy of each photo in both places, right? You have the same information in both places. If you're looking at Google Photos, you can look at any of those photos, but it's the same information. But maybe on Google Photos, you only look at the pictures of your cat. And on your phone, you only look at the pictures of your pet parakeet, but you've got all the pictures both places. Is everyone cool with this? We're fine here? Most cell division, you're keeping the genetic material as identical as you possibly can. So far, so good? Oh. Coming up, we're going to go through the vocabulary portion of today. If you get a cell, boom, cell, and you go and you look at all the DNA inside of it, that's called its genome, alright? So if you've got, like, something prokaryotic, like some little bacteria buddy, you've got E. coli, you go and you look at E. coli. E. coli has, so prokaryotic cells in general have one molecule of DNA. So they've got one piece of DNA, it's nice and long, it's coiled up in kind of this like blob thing. So they have one DNA molecule. And that's their genome, yay for them. Okay? In eukaryotic cells, which is most of everything else, there's going to be a lot of DNA molecules. Right? So you have one nice long DNA molecule, you have another DNA molecule that's a little bit shorter, you have another DNA molecule that's nice and long, again, you've got these different DNA molecules. Right? But all the DNA molecules together, that's the cell's genome as a whole. Yeah? Everyone's cool with this? And we can look at it right here, this purple stuff. Those are chromosomes. Sorry, that's genetic information. This stuff, genetic material is housed in chromosomes. So you've got this stuff that makes chromosomes is something called chromatin. You've got your nice long DNA molecule, which is long and thin. Think fishing line is thin than fishing line, but think fishing line, right? And it's associated with, next to it, there are a bunch of proteins. So you end up with proteins in DNA, and you can wrap them down into chromosomes. So every eukaryotic species, so anything that's got membrane-bound organelles, they're going to have a certain number of chromosomes in each cell nucleus. So the number might be 2, that number might be 48, that number might be 26, but it's going to have some characteristic number of that organism. Now, the cells in the organism that don't reproduce, non-reproductive cells. So that's things like kidney cells, liver cells, eyeball cells, face cells, whatever, for you, right? They're called somatic cells, alright? They have 2 sets of chromosomes. They have 2 full sets. So you have 2 sets of chromosome 1. You've got chromosome 1 that you got from your mom, you've got chromosome 1 that you got from your dad. You have chromosome 2 that you got from your mom, you have chromosome 2 that you got from your dad. You have a double copy of every single chromosome that you have in your body. With the exception of sex stuff, but we'll talk about that later. So far, so good? Cool. Those are somatic cells, and those are most of your cells. If you just randomly pull the cell, you get a somatic cell, like always, because there's billions of them. The one exception to this, are gametes. So gametes are the reproductive cells. Gametes are going to have they're going to have half the chromosomes that the somatic cells did. Is everyone cool with this? So somatic cells have 2 sets of chromosomes. Gametes only have 1 set of chromosomes. So, biological males, you're expecting sperm. Biological females, you're expecting eggs. Everyone's with me here? Sweet. So, to get this done, regardless of how you're doing it, to get this done, to divide cells, you're gonna need to replicate that genetic material. So you want to go from one cell that has this genetic material to two cells that have the same amount of genetic material as before. So what you're gonna do is that DNA is gonna get replicated. The chromosomes are going to condense, bless you, which is basically just saying, oh, instead of that fishing line mess of everything, you're gonna put it in some place that you can actually handle it. And here's a picture. Have you seen this picture before? I both love it and deeply dislike it. I like it because it's a good picture. And what's happening here is there's a really, really long DNA molecule, super long DNA molecule, that's been replicated. So this little molecule, there's a copy of it right along here. They should be identical, okay? So that top arm and the bottom arm should be identical to each other, okay? They're held together there, so call it the waist and centromere. They're held together there, but the information here and the information here should be super identical, okay? I like it for that. So this originally there's just a chromosome. It's now duplicated. Now there's two sister chromatids. The information's there. I dislike this picture because this is the picture everybody sees with chromosomes, and it's not normal. All right, normally a chromosome doesn't have that little X shape that like one molecule with another molecule stuck together. It normally doesn't have that. Normally it's just one long thing. I mean, it can be slightly condensed, but it's kind of long, stringy, all over the place. It's this thing going on here, like one of these little guys over here, this whatever random strung out shape it is, right? This, when you see it like, when you see it condensed, this is getting ready to divide. So normally it's not like that. Normally it doesn't have both sister chromatids there, and normally it's not as crunched down. Okay, just for the record. But what's going to happen during cellular division, and so before this molecule, it replicates, so now it's two of them, and now you have two sister chromatids stuck together. During cell division, those two duplicated parts are going to pull apart, and they're going to move into separate nuclei. And then, right now, when they're together, they're called chromatids. When they're separate, they're going to be called chromosomes. So it's going to look like this, and we're just looking at, it's a bad picture, but we're just looking at one right now. We have this organism that, for whatever reason, only has one chromosome in all of it, okay? Just roll with it here. One chromosome, one DNA molecule, right? It gets duplicated, this chromosome gets duplicated, and now you have two sister chromatids. They should be identical to each other. You have two DNA molecules. And then you split those apart, bloop, and you now have two cells, each that have the same genetic material, or the same genetic material as the one that was in the original parent cell. The first cell that splits, that's called the parent, and then what it splits into, they're called daughter cells. Okay? And this is so not drawn to scale, because, well, okay, has anybody here made bread rolls before? Like, if you're cooking, you're making for Thanksgiving, whatever. Can somebody act out for us? You have a blob of dough. You're gonna shape these little dudes. What do? Yeah, okay, so I don't know if you can see them all over here. You got the blob, bloop, right, and then you do whatever else you do to it. Make a decorative swan, or whatever it is you're doing, right? Okay. If you have a blob of dough, and you split into two parts, how big is each part? Are we all okay that's half the size of the original one? Is everyone okay with this? Okay, everyone's looking at me like it's obvious. It is obvious, but people get confused sometimes because every drawing in every textbook I've ever seen makes it look as if this cell and these cells are exactly the same size, and they can't be because this cell right here, there's a certain amount of stuff, and it's split in half, and I have two of them. They're smaller than it was just a second ago. Good news though, each of these cells has a full set of the DNA it needs, so it can grow and get chubby and do all sorts of things, and that's fine, that's not a problem. Okay? So these cells are going to grow some more, and that's cool, but they have the same amount of genetic material as this one did up here. Have I lost anybody yet? Great. This whole thing that's going to be splitting these apart, so we're going to stay with eukaryotic cell division. On Wednesday we'll see prokaryotic division. But eukaryotic cell division is going to have what's called mitosis, which is the genetic material gets pulled apart. And then cytokinesis, which is the cytoplasm gets broken. I actually have a slide. Yeah, this slide, yeah. Remind me what cyto means. We've had cytoplasm, we've had cytoskeleton. Did you say cell? Yeah, right, it means cell. So this is going to be cell, the like actual physical bloop pulling it apart. Okay. Normally expecting two identical daughter cells, there is one weird version that's going to be called meiosis. Meiosis is going to make gametes, and those cells are going to have half the amount of genetic material as the parent cell, and we'll get to that later, okay? But everything else, mitosis, is going to be the parent cell and the daughter cells will have the same amount of information. Okay, that was thing one to know. Thing one to know was most cell division results in genetically identical daughter cells. Thing to know number two is the mitotic phase, the actual bloop, pulling apart phase, is going to go back and forth between what's something called interphase in the cell cycle. So, for any study, we're going to look at this picture right here. And there's a lot of text in the slides. This blue thing does not exist inside the cell. There's not like a little tiny alarm clock inside the cell, okay? It's not like a physical thing that exists, right? This is a diagrammatic representation of the cycle a cell goes through. We okay with this? Yeah? So, what a cell goes through is it starts as its own little cell, right? Yay for a little cell. When the cell's just like being a cell, right? It's just growing and it's doing its thing and it's sending signals and it's spitting out stuff and it's responding to chemical signals and it's doing whatever it's doing, right? It's being a cell. That's cool. It's still in the cell cycle. The cell cycle has two main parts. It has this part that's usually about 90% of the cell cycle called interphase. And then it has a short phase called the M phase, the mitotic phase. The mitotic phase is when you're having mitosis and cytokinesis, where you're pulling apart the genetic information and splitting that cell in half. Interphase is when the cell, interphase, all this stuff, is when the cell's growing. Part of it's going to be copying the chromosomes, it's just doing itself stuff. Interphase itself, this big part, is divided into three portions. The first thing is called G1. It's for gap 1, or the first gap, right? So G1 happens first, unlike photosynthesis, where you have photosystem 2, then 1, but this G1 happens first, right? First gap. Then you get to the S phase. This is where DNA synthesis is taking place. So during any time in interphase, the cell can, as a whole, be growing. It can make its membrane be bigger, it can make more proteins, it can do all sorts of things. The only part you're expecting the DNA to be synthesized, to be replicated, is here in this S phase. Are we cool with this? Alright, so the cell as a whole can be chugging along doing stuff. DNA synthesis is happening here in the S phase. After the S phase, There's the second gap, it's called G2. And then after G2, you go into the mitotic phase. So mitotic phase is mitosis and cytokinesis. Are we okay with this? Great. This is the part that, if it was a normal year, I'd make people come up here and act it out, but I have not, for the life of me, figured out a way to act it out while they're sitting there, not next to each other. So we're just gonna go through our pictures. Does anybody think you act this out in high school? No? I don't know what people are doing with their lives. Okay, so last year made people act it out, and I'm not sure anybody, like, learned anything, but it was interesting. Okay, so we're gonna start, alright, so, staying focused. Cell, chilling, doing the cell thing, growing, getting nice and chubby. Here, genetic material got copied, and the cell's like, you know what, I need to get going and divide. Gets here to mitosis, it's gonna pull apart the genetic material, split the cell actually in half, and now you have two cells. And they both can go through their own cell cycles, okay? The G1, S, and G2 phases are nice. We're focused right now, we're gonna focus on this N phase, okay? So we've got pictures here. We're gonna start from G2 and walk ourselves through cytokinesis, all right? So, just to orient you to what's going on here. The phase that you're in is right here. You're looking at DNA is stained blue up top. So down here at the diagram, up top is actually a microscope image. It's been stained, so you know DNA is stained blue, and the centrosomes and then mitotic spindles are stained this like greenish color. Okay. So G2, the cell had been just chilling, right? You see the chromosomes kind of strung out inside the nucleus, right? Just they're all over the place. The nuclear envelope is still there. You'll note it's there in this nice like little packet. The centrosomes are hanging out here. Nothing's really happened yet. This is G2. Prophase, the cell's getting ready to go. You start forming up what's called mitotic spindle, and the DNA inside the nucleus realizes, oh, we better get organized. So it's going to start condensing down. Alright? Prometaphase, and so you can start seeing condense down here. It's a little bit tricky to see. I had fun not touching that mouse. If you don't notice this? It wiggles around. Prometaphase. Start to get organized. DNA has already been synthesized, right? Got synthesized back in S phase. So you already have the double copy of every chromosome, and they're stuck next to each other as sister chromatids. We're cool? Prometaphase, you're now actually really getting organized. You've got this picture up here, you've got, you can see right here, you can see all of our little chromosome buddies floating around all over the place. At this point, the nuclear envelope is mostly in chunks now. You have your chromosomes inside your nuclear envelope. If you need to get at those chromosomes and pull them apart, you need to be able to break that nuclear envelope down. So that's prometaphase, additional getting organized. Metaphase is the easiest one, in my personal opinion, to see. At metaphase, you have the spindles on both sides, and you have all these chromosomes lined up down the middle. It's called the metaphase plate. They're lined up down the middle. Anaphase, it's actually kind of pretty. Anaphase, those sister chromatids start getting pulled away from each other. Goodbye, buddy, right? And so, like, this cell we're looking at here in anaphase, the parent cell had 3 pairs of chromosomes. So the parent cell had this long chromosome and had 2 copies of it, so this one here and this one here. It had this medium-sized chromosome, so this one, and then this short chromosome, this one and this one. And so one from mom, one from dad, one from mom, one from dad, one from mom, one from dad, right? When it was copied, all of these pairs of chromosomes replicated, so they each had an identical body. And now that it's getting pulled apart, so once we end up with these two cells, each daughter cell is going to have these 3 pairs of chromosomes. You see that? See okay? Okay, so that's anaphase, where it's getting pulled apart. And then the last stage is actually just like putting everything back to normal. We've got telophase and cytokinesis, basically the cells now pinching apart, right? So now you have 2 cells, and you reform the nuclear envelopes, you put things back to normal. We're cool with that? We have a video, online people will, go ahead and search for it. I was going to embed it, but I'm worried about the size because there's a lot in this particular PowerPoint. Search on YouTube for Bioflix mitosis, and it's there. So go ahead and watch that one. For us, it's right here.
This soccer
And so I'll just back over.
player's knee will be as good as new in a few weeks, thanks to mitosis, a type of cell division that generates new cells for growth and repair. Let's move into a cell to witness the events of mitosis. Before a cell can divide, it must first duplicate the chromosomes stored in its nucleus. During chromosome duplication, several bubbles open up along the chromosome. Each bubble grows until it merges with an adjacent bubble. Each chromosome now consists of 2 identical copies called sister chromatids. Getting closer, we see that each sister chromatid consists of DNA wound around small proteins called histones. The sister chromatids begin to coil into tight helical fibers. Outside the nucleus, centrosomes that duplicated earlier move away from each other to opposite sides of the cell. Microtubules extend from the centrosomes, forming the mitotic spindle. Back in the nucleus, the DNA forms loops, becoming more compacted. These structures fold back on themselves, eventually condensing into a shorter and thicker chromosome consisting of 2 sister chromatids. As the chromosomes continue to condense, the nuclear envelope breaks up. The array of spindle microtubules is now extensive, and the chromosomes are fully condensed. Spindle fibers from each pole attach to protein structures located at the centromere of each sister chromatid. As the chromosomes are bound by spindle fibers from opposite poles, they move first one way and then another. The counteracting forces of the spindle eventually cause all the chromosomes to end up at the center of the cell, as if arranged on an imaginary plate. The sister chromatids are released from each other, each becoming a full-fledged chromosome. They are moved toward opposite poles of the cell, pulled along the spindle fibers attached to them. At the same time, overlapping spindle fibers that are not attached to chromosomes continue to lengthen, pushing the poles farther apart. Once the chromosomes arrive at their destination, they become less condensed. 2 new nuclear envelopes form, completing mitosis, the division of one nucleus into 2 genetically identical daughter nuclei. The cytoplasm divides by the process of cytokinesis, forming 2 separate daughter cells. In your body, millions of cells divide every second, providing new cells for growth and for repair of damaged cells.
Cool, we're good? We'll see more of this later. I was telling the 9 o'clock class that this, was I telling you? I can't remember who I tell because of me. Who did I tell you this is my least favorite chapter? Did I tell you this?
Humans produce
Stop!
gametes,
Stop!
eggs, and sperm.
They do, I know,
the
but
process of meiosis.
that's very nice. Okay. But it's really important. Okay, this stuff they're going over. Um, what happens if you've got a cell, right, and it's supposed to divide, but it just doesn't feel like dividing, and it just doesn't divide? What happens to you? We're gonna stay with you. Same deal goes for whatever kind of slime mold or chicken or anything we're looking at. But your cell's been damaged. Um, you want your good cells to divide, but your good cells are like, I'm not feeling it. What happens? Huh? You get cancer if cell division's wrong. Yes, that's the next question. What happens if they divide and you're not supposed to? Is keep going, and then they're growing, and then you don't want them to be there. You're like, stop it, right? It's growing, and it's not supposed to be there. You're like, knock it off, right? Yeah, it is so absolutely cancer. This cell cycle, the cell division stuff, it's gotta be tightly regulated because if it goes for the gusto, and say tissue just starts growing that's not supposed to start growing, that is so absolutely cancer, and it's very bad. Are we okay with that? Sorry, it's a technical term for cancer. Very bad, right? Okay, so you don't want this to go too much, because then you got cancer. You also don't want it to just not do anything, because like if you get wounded, you're hurt, or as you're growing and things changing, you need the cells back. Because otherwise, you're gonna like run out of you and be injured and bad things. Okay? Like, um, like osteoporosis, part of the problem is um, cellular materials being broken down faster than it's being built back up. Right? So it's a problem. Are we okay with this? Other things, how I think of things that can go wrong here. What happens if this whole cell division isn't right? So you have a set of DNA, and you copy it, right? You got a copy of the DNA, and now when you pull this apart, what happens if you mess up the pulling apart? What happens then? Problems. Right? Okay, that there's problems. Okay, so briefly, without going too much detail, you've got um, a set. We're gonna go with one because I have to use my hands. I'm sorry. You got, let's say, one chromosome here. You've replicated it, so now you've got two sister chromatids. You're supposed to pull them apart and have one in each cell, right? If you do this wrong, or if your cell does this wrong, and you say pull both of them to one side and have the other one not have it, this is a problem, because this cell that doesn't have that genetic material can't get it back. It's not coming back. Like, it's gone. The cell has no way to be like, oh, I feel like I'm missing an entire chromosome. Besides, like, I guess I'm not going to make any of the proteins that are coded for on that chromosome. Do you see that? This is a problem. Also, the cell over here that has all the chunks of stuff has more than normal, right? And so that's a problem too. It's more of a problem on the other side, but both of them are problems. Are we okay with this? Yeah. Okay. Um, Pictures one at a time just so you can go through it. As you're memorizing this, it goes PMAT, so G2 phase, metaphase, the P's, then MAT. So we've got prophase, pro-metaphase, right? So prophase is first, pro-metaphase is moving towards metaphase. Metaphase, they're lined up in the middle, that's how I remember it. Anaphase, they're pulling apart, and then telophase and cytokinesis, bloop, cut apart. Just so you actually see stuff, this right here, we're at metaphase, right? You can totally see these little microtubules grabbing on to the chromosomes. You see that they grab? They grab on to this thing called the kinetochores. So you can see these little microtubules coming on in here. They grab those proteins, and then those microtubules. Oh yeah, these are microtubules, by the way. These microtubules, they're gonna grow, and as they grow, so oh my goodness, I need another hand. You've got your two sister chromatids stuck together, right? Stuck together right here. You've got microtubule on this side, microtubule on this side, actually a lot. you've got them being held right there. They're being held tight. Cool? Okay with this? Meanwhile, you've got these microtubules over here that aren't actually holding on to the sister chromatids. They're just over here, right? So these ones are each going to grow. So there's going to be more here, and they're going to push out, right? So these ones are growing, and these ones aren't really feeling like growing. The end's grabbing on to the sister chromatid, and they're not growing. But these ones are growing. So one pushes out, and the other one stays the same length, which means they tug, and then it's going to break. Let's see it here. So as these ones each keep getting longer, so they push the cell apart. These ones didn't, so the sister chromatids get yanked apart. Do we see that? Are you up for one concept more? A short concept? Short concept? Short concept is this. It's called cytokinesis. Where you split it apart. In animal cells, it's called cleavage. Cleavage just means cut, by the way, just so you know. You got this animal cell, you got a cleavage furrow coming in, it's going to split apart. You end up with two daughter cells. In plant cells, you do it slightly differently. You pull apart, so for your daughter cells, you pull apart the genetic information into two different blobs, right? In plant cells, you form up a new cell wall right along the edges, and that effectively splits it in half. Do you see that? Great. Questions? Comments? The most exciting chapter you've ever seen? It does set this up for all the genetic stuff, which people typically like, because they're like, okay, if I marry somebody who's got red hair, purple eyes, and 12 toes, my children, and then they, whatever. So that's what I've got. Do remember to do your lab in some form or the other, and if you need stuff, talk to your lab person. I think that's it. Oh, and I'll send a study guide for